million deaths worldwide each year. They're the 6th leading cause of death across the globe and the number 2 leading cause of death in the United States. So it's a really big deal. And it costs the government $10.5 billion a year for funding and upkeep. So before we get started again, we need to talk about some terms. So the first thing is NSCLCS, or non-small cell lung cancers, but I think it's easier to say non-small cell lung cancer, so I'll refer to it as that. TKIs, or tyrosine kinase inhibitors. Basically, an inhibitor, in this case, is going to inhibit the tyrosine kinase, which activates things, so we're going to block activation. And MET inhibitors we're going to talk about as well, which are also referred to as HGFRs, which are hepatocyte growth factor receptors, which also trigger cell growth. Really important in cancer, obviously. Does anyone have questions about these before we move on? So, what are these researchers trying to do? There are 2 main ways that cancers develop resistance to drugs. First is through mutations, right? Cancer, by definition, is uncontrolled cell growth. So the checkpoints that make sure that the cell is not rapidly mutating get bypassed, and you get mutations. So if we're trying to administer drugs, that's a really big problem because the cell keeps changing and we can't keep administering the same drug. The second thing that they focused on in this paper are these bypass tracks. Bypass tracks are essentially when the cell finds a way around the area that's inhibited by the drug, and then farther downstream you get cell survival. So that's really bad as well. And in this paper they also did something cool, which is trying to find a way of harvesting cancer cells from patients to use and study how they developed their resistance, because what's happening is researchers are going into the lab and they're growing up these tumors that they know are going to have resistance to the drugs, but they don't know how the cells technically develop this resistance, and so they can't use those results in clinical settings. So by taking cells from patients, they know that that's actually a viable way of saying, okay, we can apply the results of this to other people. Does anyone have questions on this? So basically, how do they do it? They take a person with these non-small cell lung cancers who has been treated with these tyrosine kinase inhibitor drugs and who's developed resistance over time. They biopsy their cells, their tumor, and they grow it on a petri dish so that they have their tumors. That whole thing on the right here, is basically a control just to show you that the cells are viable after 92 hours. So don't worry about that. Okay. So the next thing is, what kind of drugs they focus on. So they went through and they ran this test called GI50. It's complicated, but essentially what they wanted to see was that if there are these spikes, it means that the drug inhibited the bypass pathway, which is what we're trying to focus on, right? So these spikes are really good. And they found that only 2 groups had these spikes, these MET inhibitors, and this other drug they didn't mention, but it was a one-off, so they decided to focus in on these MET inhibitors.
Yeah, I don't understand
It's this process called GI50, and essentially they measured the levels of this chemical later on, and if it's present downstream, it means that it inhibited the bypass pathway. So higher spikes mean higher levels of this GI50. Higher levels of this GI50 mean stronger binding affinity to the bypass pathway. Does that make sense? Anyone else? Yeah.
So each column is a different cell line?
It's a different type of drug or inhibitor they've tried, yeah.
Oh, on the same cell line?
On the same cell line. Any other questions? So we're focusing on MET inhibitors now, and they picked 2 MET inhibitor drugs to work. Crizotinib and geftinib, and funny names, but what we see here with this red line is that the drugs are each effective on their own. We see that what they're measuring here is the number of cells, so if the red line is going down, that means that the cell count is going down, so the drugs are effective in stopping tumor growth. So that's what these 2 are showing here. What we see here, these are the cells, the tumor cells on a petri dish. This is our control. This is the first drug, geftinib. This is crizotinib, the second drug. And this is the combination of the 2 drugs. And it's really obvious, you can kind of see. So these all have tumors, and it's kind of hard to see that it's reduced. But we have no tumor here, which is huge. They found that the combination of these 2 MET inhibitor drugs kill the tumor, which is really, really big. So then they said, okay, well, if these two drugs work, what else works? So they focused in on these two other MET inhibitor drugs called AZD6244 and ceritinib. And again, we're seeing here that the two drugs, this is ceritinib and this is AZD6244, and they're both effective on their own again. But of course, we're not interested in how they are on their own. We want to see how they work together. So when we did this, this is the best drug combination they found because this is your control here. Then you have ceritinib, one drug. You have AZD6244 here. But when combined, you have a negative, which means that not only did you stop cell growth, but you actually reversed and started to shrink the tumor, which is huge because not only are you not causing the tumor to keep progressing, you're actually shrinking it and causing the cancer to start to go away. And you can see this is the same thing here. This is about the rate, the tumor volumes, and it's shrinking, and it's all below below the zero on the x-axis here. And then this is cool. This is kind of a picture. So this is the lung, and here's where the tumors are talking about, and you can see here that the tumor's basically gone. Does anyone have questions on this? Yeah, Andrew.
So the change in tumor like this that causes the shape of the combo drug, when you meditate down, it goes back up, of course. That's because the tumor is dead, not because it's developing resistance to the combo.
Exactly. Yeah.
How did they decide what drugs to combine?
So they know of a certain number of these MET inhibitor drugs that are effective because they've been using them before. They took well-known ones they know are effective on their own, and then said, okay, well, what happens if we start to pair them together? Yeah.
Did they use that connecting drugs with the original drug? And it like resensitized?
Yeah. So that's what we're going to get to is after this, they found that not only did it shrink the tumor, but it resensitized the cancer to the original tyrosine kinase inhibitors. Yeah, D.
You may have just said this, but after the tumor volume was reduced 100%, how does it continue to reduce? So the red line is graph out.
This,
That's the combined treatment, right?
Yes.
So, okay, so we're going to say there's a 100 gram tumor.
Okay.
As it goes from 0 to 100%, negative 100, that tumor is now not there.
Right, which is why the line goes basically back up to 0 because there's no tumor to be shrinking anymore.
But on that, that can't be because right. So if it drops to 0, the tumor size is 0, and then it is at some point again 50% and 25%. So it's just decreasing in volume slower. Is that just because this tumor is so?
I don't.
Yeah, I don't get it.
I understand.
Yeah, it's basically the tumor is ending up smaller than or negative.
That's the point, right? Is it ends
No,
up
what
smaller
he's
than.
saying by that, it's like you're basically creating like a negative tumor.
Oh, you're
I
saying
know.
because it goes to zero, negative 0% volume change and then back.
Does it not actually go to minus 100? Is
No, it's just.
okay. All right.
Any other questions? Yeah.
If each of these MET inhibitors are targeting the same protein in the pathway,
Yeah.
what's different about them that increases their effectivity other than just using a greater volume of them?
Right. So what we don't know is we know that there's a bunch of bypass pathways. We know that they find ways around our drugs. But the question is, which pathways? How does that work? Where are they, changing? And what they found is that there are these two main pathways that they tend to go around specifically for these tyrosine kinase inhibitor resistance. And so they've targeted these two pathways, and the drugs that they've used, like the AZD6244 targets one pathway, and the ceritinib targets the other pathway, and they found that in tandem it is really effective. Any other questions? Okay. So, basically, our results again are that we stopped tumor growth and shrunk the size of the tumor, and we led to the resensitization of the cancer cells to the original tyrosine kinase inhibitor drug, which is really important. And the biggest thing that you get away from this paper is that each of these treatments and these drugs are used from specific patients' tumors, meaning that we're developing these drugs and knowing that these two drugs in tandem work well for this person, which is really, really important because we can now figure out, okay, well, if we can know that these two drugs work well for this person, what if we get a biopsy of this person's tumors, and we look at them and we find out that these 2 drugs are effective? Well, then we can go tell the doctors, these 2 drugs are going to be your most effective bet at treating this person's cancer, and then they can administer those drugs to the person. So we're now customizing cancer treatments. Yeah.
How long would it take to biopsy that and figure out which one was best given that time to get the cancer progress?
Absolutely. I mean, the experiments themselves to test these things took about a month or so for each set of tumors they went through. So that's clearly a long time. So if there's a patient sitting in a hospital bed, they can get worse definitely in that time. But I think the point is that it kind of hopefully would outweigh the costs, so that if they're sitting around waiting, yes, but they know that when they get the drugs, those are going to be the most effective drugs. Liz?
Do you think this kind of specialized cancer therapy is practical at this point in time?
So, I mean, I think it still has some kinks to be worked out, in that they need to make sure that it can work for a broader spectrum of things, because in this paper they focused on 2 types of drug-resistant cancers. So I think once we can kind of broaden that spectrum, it'll be more effective and it'll be worth doing it more, because I think only we're doing it for 2 type of resistant cancers.
How expensive is this type of treatment? Because even if you do have that small lung cancer specific, are you going to be able to have access to this type of treatment?
Right. We don't actually know, because they just did them in a lab setting, so they didn't actually go back and administer drugs to patients. They just biopsied their cancer cells and then worked on them in the lab. I presume it's going to be a costly thing. I mean, chemos are. But I think the point about this was that we can say with more certainty which drugs are going to be effective, instead of just guessing and hoping that regular chemo is going to do the trick.
Say you are a patient and you send off your biopsy or whatever, you have to run the test. While you're waiting, would you have to sit and take no drugs and risk changing the way your tumors are, which will change the way the drugs work, or would you be able to take
I presume yes. They could probably keep trying to administer the original drug that your tumor developed the resistance to, and hope that it's doing something maybe. But I presume if you start putting other drugs in, you can get different resistance, and then the combination they come up for you won't be the most effective. Yeah, Andrew.
You mentioned that they talked about how the drugs desensitize cells to the original tyrosine kinase. Did I just miss back on that?
No, there was. They basically, that was one of their points they talked about. They didn't have a graph that actually showed that. But yeah.
If a cancer, if a sample of a tumor is mutating separately in a petri dish differently than how it would be mutating over that month-long period inside the person's actual body, I don't know if they addressed this, but just based on the rates of mutation, do you think that when they find a treatment that works for the sample, would the cancer be different enough in a human body that it would be near as effective?
Right. I think by that point, your mutations in the general sense that just the cells rapidly dividing, so there are those kind of mutations versus these specific, bypass pathways, you're going to get the regular mutations just because that's the nature of what cancer is. But I don't think they'll start forming new bypass pathways, because as long as you keep administering the same drug over and over again, that's the only pathway. They're going to keep going around that drug because it works, right? Don't fix it if it isn't broken, right? Any other questions?
Why did you ask that finance question? Where did that come from?
I was curious just because customized medicine isn't always cost effective in other treatment, so I don't see it really being sort of piggybacking on the practical question.
So that's a huge topic right now in cancer research. The cost of cancer therapies are incredible. Sanofi, there's a news report this podcast called Radio Lab that was talking about a $40,000 cancer treatment that extended your life by 40 days. And it comes down to a very kind of fundamental question, but when we hate as biologists to consider, but what is the actual financial, what is the true value of a day financially in our lives? Because once these treatments, once they're FDA approved and they're approved by healthcare companies, you can't really deny them to people in those situations. It's absolutely fascinating. You guys know the hepatitis story? Okay, so yeah, I'll just leave you with this, but there's a cure for hepatitis. And if everybody with hepatitis gets the cure at the same time, it'll basically bankrupt the system. It'll bankrupt healthcare. So they have to think about who gets this cure, and it's not a treatment. It's a one-time cure. So once you get this over the course of 10 months, you're done with hepatitis. Well, certain type of hepatitis. But if all of it hits at the same time, we're done. So it's kind of an interesting limit that we're now hitting, and that's why I was asking if you got that.
I mean, just looking for working on using the same method for other types of cancer, not just lung cancer, and not just the 2 types of resistant cancers we talked about. So I just wanted to say thank you to my parents, Catherine and Giles, and my sister Ellie for letting me drive you to school every Thursday, crazy early. Dr. DeCilio, Mr. D, Mr. Maxwell, the whole Journal Club team, specifically Catherine and Amanda and Sydney for helping me with my presentation.
Next week is Liz, so come.
IRT, I'll see you tomorrow at 7.